So hello everyone, and thank you for coming to this evening's BMS Talk. It's my pleasure to introduce our chair for this evening, Christian Taylor. Christian works at the Open University's School of Environment, Earth and Ecosystem Sciences. He also teaches science to school students by running COVID catch-up classes on particular science topics that students missed during the pandemic. Christian runs fungal forays on Dartmoor and in Devon during the autumn season. And at the moment, he's establishing an ecology and sustainability education project in rural France. So thank you very much, Christian. I'll hand over to you.
Okay, well, welcome to tonight's session from the British Mycological Society. Tonight, we're going to be hearing about lichens. And as you know, lichens are at least half fungus. So we're going to be hearing from Fay Newbery, who's going to be talking to us about Lichenicolous fungi, which are fungi which actually involve themselves and relate to already existing lichen colonies. So if you like, a third fungus or a second fungus comes along in addition to the one that's already involved with the lichenization event that happened previously. So Fay Newbery, if I just introduce her, she works as a plant pathologist, diagnosing diseases on garden plants. And during the summer, Fay works at the Royal Horticultural Society, helping the public to diagnose their own unhealthy plants. She's particularly interested in diseases caused by fungi, both in UK garden plants and in our wild flora. The obvious link between this work and her interest in lichens was to begin looking at fungi that grew on lichens, that is the Lichenicolous fungi. So today's talk is called Lichenicolous fungi, an expanding area of mycological study. Thank you. So, well, I'll hand over to Fay, and what I will be doing is looking at the chat area. And if you wish to ask a question or type a question at any point, please do so in the chat. And then at the end of the talk, I will be fielding some of those questions to Fay in the question time at the end. So I think
right, ready, Christian?
Yep. Okay, I'm going to turn off my video and we'll hand over to Fay. Thank you, Fay. Wonderful, thank you.
Right, well, welcome everyone. I'm just going to sort my screen out because I've got that wonderful Zoom bar in a place where I really don't want it. Now I'll try and move it. There we go. It's out the way now. So I wanted to begin by thanking the BMS for inviting me to talk today. But I also wanted to thank particularly a whole group of people who allow their photos to go on the internet with Creative Commons licenses, because I have used a lot of photos that are not mine. If you were restricted to seeing my own photos, I wouldn't have been able to make this talk. So thank you to everyone who is kind enough to do that kind of sharing through Creative Commons. Perhaps surprisingly, fungi that grow on lichens, or Lichenicolous fungi, appear to be attracting an increasing amount of interest, although maybe that's just among lichenologists. More and more species from field trips have lists that include Lichenicolous fungi as well as lichens. And each year we find more Lichenicolous species in the UK. David Hawksworth has been interested in Lichenicolous fungi probably for longer than anyone else in the UK. He saw them as a source of yet unexplored biodiversity. And you can see from an image from one of his papers that even the spore types show an amazing amount of diversity. So what are Lichenicolous fungi? They are fungi that live on or inside lichens, but they're not the fungus that make up the microbiome part of the lichen. So they're not essential to the lichen symbiosis. They're not doing that job of forming the lichen. They're an extra piece. There are other fungi which have been found in lichens now, which usually live as yeasts and do seem to be absolutely necessary for the symbiosis. We're not talking about those either. Very hard to even tell that they are there. They've been found by molecular methods. I'm going to be talking about Lichenicolous fungi that grow on a lichen or in a lichen, but we can see signs of them. So usually that's because they pop out on the surface at some point. The fungi on lichens have many different relationships with the lichen they're growing on. Some are definitely pathogenic. That is, they take nutrients from the lichen and they harm the lichen while they do this. They can be found on lichens in various states of healthiness, from normally healthy, nearly normally healthy to dead, depending on how much harm the disease has done so far. In this photo, you can see Didymocurtus slaptoniensis. It's not only turning the lichen that it's on red, but it's preventing the lichen's own fungus from producing spores. So it's definitely causing harm. So if you look inside, you can see in Mark Powell's middle photo there that the spores are coloured red. That's where a lot of the red colour actually is inside the lichen, because of this fungus being present. But if you look at the shape of those spores, and what you're seeing is two little parts making one spore, because the dye has coloured the internal parts, not the cell walls. If you try and compare that to the photo of Paul Cannon's ascus at the side, you can see that those spores are misshapen. I very much doubt if they're viable. Other Lichenicolous fungi appear not to interfere with the growth of their host. This fungus I've found lots and lots of times. It's really common. You can find it on healthy hosts or stressed hosts. But I've found one where a Vauxhalla pycnidium was actually growing side by side with the pycnidium of the host. And that pycnidium of the host seemed to be producing undamaged spores. And to me, that suggested that this Lichenicolous fungi gets in there, steals its food from the lichen, but it doesn't cause a lot of damage. It's possible that there is mutualism going on, that some of these Lichenicolous fungi grow on the lichen, they don't do any harm, and they actually do some good. So the lichen maybe likes having them. But whether this actually happens or not, I don't know. Fungi are heterotrophs, so they have to feed from something. They can't make their own food. That's why a lichen forms in the first place, because fungi that have lichenized have got themselves a photobiotic partner that can feed them. So in order to be a mutualistic Lichenicolous fungus, you are going to have to take food from the lichen host, but you're going to have to give something back. And so far, we don't know whether that happens. But it could be that there are fungi that live inside the lichen that we don't notice for a long time. We tend to suggest the name endolithic for those, and they're equivalent to the endophytes that we know in leaves. And in the case of leaves, those endophytes often produce sporulating structures as the host leaf dies. So maybe what we see as saprophytes coming out of harmed lichens are actually endolithic fungi that have been in there doing some good for a long while, but when something has happened to the lichen host, they then can't feed from the lichen anymore, and they use the dead lichen material maybe to sporulate. How would we know? And then you've got things that we are pretty certain are saprophytes, which are feeding on dead organic materials. So they're not feeding on the lichen until it is already dead, and they didn't kill it. Many people who study Lichenicolous fungi like to ignore the saprophytes. I think there are two reasons for that. Sometimes there are very few fungal characters, so successful identification isn't always possible. Another argument for excluding them is that many of the saprophytes we find on lichens, on dead lichens, also eat other dead material, so they're generalists. But there again, if we don't record the fungi that we find on lichens, even when we think they're saprophytes, how would we ever know whether they're generalists or whether they're limited to living on dead lichens? So I tend to look at everything. This photo on the left shows a fungus that had a dead area. It was a punctilia species, so a green leafy lichen on a tree, but it had this dead area for some reason, and it had this sort of pink kind of pinkish glow to it. And when I looked, I found the hyphae and the conidia, which you can see on the right. And a thorough search of the living surface of the lichen didn't produce any of those hyphae or any of those conidia at all. So it seemed to be able only to feed on the dead part of the lichen. So what types of fungi grow on lichens? This for me is one of the reasons why I like studying Lichenicolous fungi. It gives me a chance to look at a wide range of fungal morphologies and look at a wide range of taxonomic groups. So rather than specializing by a taxonomic group, I'm specializing by the niche in which these fungi live. I'm not actually that interested in what the fungi are doing. Other people are. Other people like studying the biology of Lichenicolous fungi. But I like the identification process. I like trying to work out what I'm seeing and get some way through the taxonomy. The majority of Lichenicolous fungi are actually ascomycetes, but there are some basidiomycetes, and there are even a couple that could be termed macrofungi, such as this one pictured, Arenaria pultigerina. It grows on Pultigera species, that's the dog lichens. They're quite big, they like quite wet environments. As a genus, they're quite common, and they produce basidiomycetes that field mycologists would think is almost a normal fungus. The caps of these toadstools can be up to 2 centimeters in diameter, and this is probably the biggest fungus I ever get to look at. I ignore anything big. As well as sexually reproducing fungi, you find asexually reproducing forms, so even more diversity of morphology. Now, I've used some old names here, celiomycetes and hyphomycetes, and I know that these names now are anathema to people in the molecular age when we want names that show us information, give us information about taxonomy and about phylogenetic relationships. But to me, these words are useful. In Lichenicolous fungi, we hardly ever find a cervuli, but we do find lots of pycnidia. So there's a whole group of Lichenicolous fungi which have pycnidia, and to me, they are a group, because once I see a pycnidium, I can look at that group and think, right, the answer I want is there. And the same with hyphomycetes. If I look at a hyphomycete and I look at the structures within it, that gives me some words to use to look up an identification or to talk to another scientist to get some help. Generally, though, when I talk to anyone else and I say I've got a hyphomycete, they switch off. But to me, they're all equally interesting. So the first thing I'll do when I see a Lichenicolous fungi is find out as much as I can about its morphology, take photos, I'm useless at drawing, and take some notes. I don't often take measurements. For me, the characteristics of the spore shape, colour, septation, and the information about where it's growing and how it's growing on the fungus, on the other parts of the fungus, that's more important. And I never go for spore measurements unless I absolutely have to. One of the things that really helps when you're identifying Lichenicolous fungi is knowing the host. And I think that's why many lichenologists study Lichenicolous fungi. They know the host well enough to know when it's not looking quite right, to know when it shouldn't have orange spots or black spots, or to know when it's too bleached, it's too pale in colour, or it's reddened. So I think knowing the host and what it looks like is really, really important. Just as in my normal role of a plant pathologist, I know the plants, or I try to know as many garden plants as possible, so that I can see what is different and what the symptoms are that the disease is causing. In this photo here, you can see a fungus called Unguiculariopsis thallophylla. This grows on Lecanora species. It has an apothecium, but it tends to curl over itself and close up and hide its hymenium away to protect it. And then on wetter days, it will open up. It's quite hairy on the outside. This is really host specific. So if you see this, you can actually name the host. I do always check at the moment because we're not 100% sure. We used to record this fungus on a lichen called Lecanora chlorotera, but lichenologists have now split Lecanora chlorotera into quite a few different species. And this seems to be only on the subspecies, if you like. They're not subspecies, but out of that group, it seems to be only on Lichenicolous hypocarpa. But until we get lots and lots and lots of data from all over the country, we can't know whether that's true or not. And I think what we really lack is data from all over the country. Lichenologists that study Lichenicolous fungi are not that widespread, and records tend to be in specific areas of the country, which you'll see later when I show you on the map. Knowing the host also allows you to pick out the host's own structures and say this is a normal part of what should be there. And one of the things that you often need to differentiate between is the host's own pycnidia and pycnidia that shouldn't be there. And I made a big mistake in the case of Xanthoria periotina. I kind of assumed that it would have black spots for its pycnidia, that they'd be sunken and they'd show as a black dot on the surface. Turns out they don't. But that was an easy mistake for me to make because a lot of the books that describe lichens are really disinterested in pycnidia. If a lichen has vegetative structures, we call them soredia. They're little tiny balls of lichen mixed with alga, or they might have little outgrowths, which we call isidia, which break off, and they again have lichen and alga, or the other photosynthetic partner that there might be there. Lichen books talk about those, and they talk about apothecia and they talk about perithecia, but they don't so often talk about pycnidia. And sometimes all you get is the example here, where Smith, the flora for the British flora of lichens, all it says about Xanthoria's pycnidia, or asexual form, is conidia, mostly small, ellipsoid or bacilliform, colourless. So that's a genus description. So it doesn't tell me in Xanthoria periotina whether those are ellipsoid or bacilliform. They're actually bacilliform. It tells me very little. It doesn't even tell me how they form. But in lichenized fungi, it does tend to always be in pycnidia. You don't get hyphomycetes structures on the surface. So that's another thing that's important about knowing the host. So if you want to find some Lichenicolous fungi and you've never found any before, where's an easy place to look and what's an easy thing to look for? The Lichenicolous fungi that grow on the outside of lichens tend to do better in the wetter months. They're very exposed to the environment. Lichens can cope with that exposure and they can cope with periods of dehydration really well. But the fungi that grow on them can't always do that. So if the fungi are within the lichen thallus and just poking a small hole out where they throw their spores out, perithecia and pycnidia sunken in the surface do better. But hyphomycetes, some basidiomycetes, they tend to grow best in the winter months when it's wet. The other thing you can do is check common lichens, because we know that when the host population is large, such as in a field of wheat, you're more likely to get disease, and it's more likely to spread from one individual to another, so that there's a lot of disease present and it's easier to spot. And look at lichens you know, because of the fact that you know their features, and you can tell when something is wrong, when there are disease symptoms, discoloration, poor growth, and you'll be able to recognise structures that shouldn't be there. At the moment, we've got lots and lots of atmospheric nitrogen pollution in the UK. So there are three species of lichen which form a very common community in places where there's plenty of nitrogen in the air. And two of those are grey, small and grey with tiny lobes that often don't stay pressed to the surface but stick out quite a lot. Two Fischia species, Fischia ascendens and Fischia tenella. And they're easy to find. They have lots of Lichenicolous fungi that we've recognised on them, and pretty easy to work with. Most of the time, you don't even have to be able to tell Fischia ascendens from Fischia tenella, because they're so closely related genetically that the Lichenicolous fungi that grow on one tend to always grow on the other. So pinks is a good place to start. On either of those two grey lichens in nitrogen-rich communities, and you'll see that this yellow lichen is always alongside them in that community. you get a couple of pink fungi, truly pink fungi. Elosporiopsis christianseni forms pink blobs. And I call them blobs because they don't have a definite shape. They're irregular. They literally do look like wet blobs until they dry, and then they look like dry blobs and tend to go a bit darker. What you've actually got here is a sporodochium. So you've got conidiophores coming out either from the lichen or from the bark nearby. And those conidiophores are covered in thousands of coiled conidia, which you can see in the photograph from Lucas Large. He's caught them really well. They're all spiral, they're all tangled into little groups, and they sit in some sort of almost gelatinized goop, which is, and altogether, they're pink. I imagine they're rain splashed. I used to believe that if you sprayed them with water, they would appear to dissolve as the spores loosened from each other and spread out into the water. But a member of the British Lichen Society has just proved me wrong. She does enjoy looking at biology and doing biological work, and she has sprayed lots of these, and she's found that if they're really freshly formed, they might deform with water, they might spread out a little bit. But if they're not really fresh, they tend to just expand a bit when the water hits them, and then over time they dehydrate again and they go back to how they looked. So again, the books and I, totally wrong. The other pink Lichenicolous fungus that you get on these two species is Lichenicolous. This I think needs microscopic verification every time, because I have tested some of these pink, kind of pale pink growths. Or, no, they're not even growths, it's just like a stain. You can see the shape of the lichen, they just turned this pale pink. And they often look as if they're covered in cellophane or a piece of cling film. They've got a sort of sheen to them. But I've found that where I believe that it's a different fungus. So I always check latest area if I think I've seen it. And this is a Basidia mycete. So what you're looking for is very long, in this case, Basidia, that just have two sterigmata. They only produce two spores. So if you find the spores or the Basidia, you know that you've got this species. Not quite as common as the Elosporiopsis, but well worth looking out for if you're on those hosts. This is another Lichenicolous fungus that's on the same hosts. We've gone into corals and oranges here. This is Erythricium aurantiacum. This forms bulbils. It's another Basidia mycete, and its main reproductive method seems to be producing these bulbils. I can imagine a bird sort of walking along, catching them on their feet and sort of flying off somewhere else. It's very pathogenic. It's always on a part of the lichen that is very, very sick, and sometimes almost skeletonized. It can range from a pale orange, corally colour, to a very bright, almost fluorescent, right in your face orange. Usually when the more colour it has, the more sunlight it's been exposed to. I often find Basidia on here. They're not easy to find because they tend to need high humidity for the hymeneum to form. So I found that the way to look for them is to look under the lichen thalli. So you find where the fungus is because it's got bulbils on the top surface. And then if you look underneath those infected thalli, those infected little scraps of the lichen, then that's the place where you're likely to find a kind of soft looking, almost furry surface. And that's the bit to put under a microscope. And you find Basidia with 4 sterigmata. The same species, Erythricium aurantiacum, can infect Xanthoria periotina as well, even though that's a rarer host. And here you can see what the damage that that's happened in a patch of the host lichen due to this fungus. So in the photo, you can't really pick out any bulbils. I can assure you they were there. This lichen is photographed wet, and you can see that some parts are greener. And what's happened here is that the Erythricium seems to have selectively attacked the algae. So the damaged area no longer has any alga. Now, we can imagine that the lichen in that area may die because it's just not being fed by its alga anymore. Or does the erythraceum eat the alga and then eat the lichen as well? I don't know. But even when Xanthoria periotina is dry, you can still pick out a sort of paler colour, a more yellow colour to the infected patches, while the rest of the lichen is always a bright kind of orangey yellow. So that's one to look out for in that nitrogen-rich community. Moving to slightly less like nitrogen-rich communities, where you might have a lot of grey leafy lichens, you might find another bright pink fungus, and that's Mycological corallinus. It's another basidium mycete. It's another Lichenicolous fungus that grows bulbils. But in this case, basidia have never, ever been found anywhere worldwide. The fungus itself has been found on over 40 genera of lichens. So it's quite common, it's very widespread, it's not fussy about its host. It does seem to be aggressive and kill its host. But looking inside the bulbils, you can see the cells that Paul Cannon there has photographed. They're just a reasonably tight but not solidified lump of hyphae with small cells, and no reproduction going on apart from this vegetative form. The same fungus growing on a crustose lichen, so a lichen that is just like a crust on the surface that's growing on, it looks totally different. This was found on Ophiopalma ventosa, which is a lichen that I never see because it likes the west of the country and it likes high ground. And you can see that there's a wave of fungus growing out from the center of the infection. In the center, that wave seems to have almost passed, but that's where the bulbils are. And the bulbils on crustose species are often, rather on the surface, they're often a bit sunk into the lichen itself. So they're a bit embedded. And you can see that the pink is brightest at the leading edge of the wave as it moves out into fresh lichen and feeds. Again, with a coral or orange, this Lichenicolous fungus forms perithecia. They're small, they're not embedded in the host, they sit on top of the host tissue. They have a little red ostiole, they look really cute, they're very furry. And they do an amazing amount of damage to the lichens that they grow on. So here, I couldn't even tell you what it's growing on because it's so damaged. I think there's moss in there. It's not feeding on the moss, so I imagine we're trying to think of a lichen that had grown on the moss, and then the Lichenicolous fungus grew on top of that. But what you do find with Paranectria oropensis is that you get a huge amount of cobweb-like fungal growth superficially across the surface of the lichen, as well as delving down into it. It does sort of raise a biological question for me. Does that fungal cobweb have apressoria, little extensions on the sides of the hyphae that press into the lichen, or does it go deeper than that? But whether I'd ever actually work on answering that question for myself, I don't know. I'm much more interested in the identification process. But this is a cute little thing, but not cute if you're a lichen because it is so aggressive. Another species that's really aggressive and causes cobwebbing is Athelia arachnoides. Now, this fungus surprises me because it used to be really easy to find. So I used to regularly go to Leighton Buzzard to visit relatives, and I could spot this on a tree at a huge distance. So you can see Alan Silverside's picture of a tree trunk there, and you can see in the background, it's in the shopping centre. This fungus seems to like urban communities. It likes that nitrogen-rich community. That tree is covered in Fischia species. There's a little bit of yellow on it, so a tiny bit of Xanthoria there. And you can see the aggressive damage that this fungus has caused in two patches. It likes eating any small nutrient-rich lichens. It also likes eating algae. So if the tree's got algae on it, it will grow over that as well, and it will set down these cobwebs. But it just doesn't seem as common anymore. It's another bulbil former. So it forms very pale bulbils, which then darken with age. But in this case, bulbils is no longer the correct word because these are sclerotia. So these are slightly more tightly packed, and the outer layer is really, really strong and very protective. So this suggests that these would last a lot longer than bulbils, be a lot more resilient to climate, to times where the climate wasn't suitable, and dehydration as well, I guess. So yeah, sclerotia instead of bulbils in this case, but they look about the same until you actually squash one and find the skin. But you can get this fungus to produce basidia. If you take some of it and you put it in a polythene bag, you put a bit of tissue, wet tissue in the corner, close your bag up so that tissue will help generate really high humidity. Go back after a few days and have a search, and you may find basidia. But I've never found them on a fresh specimen that I've collected outdoors. So we've looked at some different morphologies already. What else is there? There are quite a lot of different morphologies and different colours and textures that you can get with your Lichenicolous fungi. So I think we've got time to look at a few more. You can get galls on fungi. Galls can be galls on lichens. Galls can be caused by nematodes, bacteria, and fungi. But amongst the Lichenicolous fungi, only the Ascomycetes cause true galls. So a gall is a proliferation of host tissue caused by the presence of another organism. And Ascomycetes definitely cause that proliferation of host tissue. But the so-called galls caused by the Basidiomycetes aren't galls at all. They're actually irregularly shaped Basidiomata. So they're the reproductive structure of the fungus that is growing on the lichen. Teligala oliveria, I can't say this, Teligala oliveria, that's the best I can do, is really common. It's really worth looking out for. You're on a common host again, Xanthoria periotina. And when this fungus first invades, it causes little flat, often three-cornered galls. Some books describe them as looking at like three-cornered hats. So I don't know whether my cursor is working, but if it is, we're looking here and here. Over time, though, the host tissue thickens and it forms space for lots and lots of perithecia, which look black. So they're embedded, but the little part that you see is black, suggesting that you've got a black structure inside there. So those black spots are the ostiole coming up to the surface, so the spores can be released from the perithecia. But what's interesting is that the perithecia aren't black. The ostiole area is blackened, but the rest of the wall of the perithecium is really, really pale. So all it takes to know that this is the Lichenicolous fungus that you've got in a gall on Xanthoria periotina is to do one cut through with a razor blade and have a look. If you can see that the walls are pale, that's it. It's the only Lichenicolous fungus with pale walls, and no microscopy is needed at all. Another gall causer on a reasonably common host, this is on Fay orbicularis, which again likes nutrient enrichment, maybe not as much as the Fiscias and the Xanthoria periotina, but it's still quite nutrient-rich trees, but often nutrient-rich bark, which is enriched by dust. And on this, you get galls caused by Lichenicolous obscuroides. And I put this one in because on the right-hand side, you can see that in this case, when you cut through those structures, it's not just the ostiole that's black, the whole structure of the perithecia are black. Amongst the gall-like causes in the Basidiomycetes, this one causes really, really pretty almost galls. They start off a kind of pinky beige and then they darken to brown. It's called Bioteropsis osnearum. It's not actually that common, but I like it. This is a totally Lichenicolous genus, with 11 species worldwide, but so far we've only found 2 of them in the UK. And unfortunately, they don't seem to be that common. So that's probably not one that you'll find. But you may well find a species of tremella. We have 20 species of tremella in the UK, just among the Lichenicolous part of this genus. The genus seems to specialize in parasitizing fungi and lichens. I've got no idea how many species we have in the UK or worldwide. It's a huge genus, but definitely 20 species Lichenicolous in the UK. This one's on a Ramolina, which is another corticulous species, likes to grow on bark. And I think they're quite nice growths, those Basidiomorphas. Again, that hint of pink. But unfortunately, the tremellas are not easy to identify. They seem to overlap in host ranges, and so that isn't as much help as you would hope. And the other features that characterize them are their basidia. And their basidia as a group have longitudinal, oblique, or transverse septa. So it's easy to say that something's probably a tremella, but then separating one tremella from the other, I find very challenging. Fun, but challenging. So let's have a quick look at another parathecium producing Lichenicolous fungus, just to show that they don't all cause galls. So on the left, you've got a photo of a very sick piece of Ramolina fastigiata. Up in the top left corner, there's a healthier piece looking sort of grey-green, but some of the host there is totally bleached, very, very harmed by this parathecia producing Ascomyces. This is Didymocurtus ramolini, so it's related to that red causing Didymocurtus on the Xanthoria that we saw earlier. But this can produce pycnidia and parathecia. They're both sunken, they're both black, they both look like little black dots. And whenever I find this species, I A, don't know that it's that species until I look inside these structures, and B, I don't know whether I've found the asexual form or the sexual form or the 2 forms growing together. So it's quite a fun thing and always needs checking microscopically. But there's plenty of Lichenicolous fungi with apothecia as well. This is Arthonia peritonearia. It's a genus, Arthonia is a genus that has a lot of lichenized fungi. So it forms a lot of lichens, but it also seems to like eating a lot of lichens. It's not always causing a huge amount of damage though. So maybe it's just getting in there and feeding from the same alga and not disrupting the actual symbiosis of the host lichen that much. Here's another Lichenicolous fungi with apothecia. I find this one interesting because in the same structures, the same black structures, which do tend to be a bit large, you find apothecia mixed with parathecia, all clumped together and often sharing the same walls. So I've managed to make a section there on the right, which shows some pycnidia, which tend to be smaller, and some apothecia. And I find that interesting. I love getting sections of that look really good. And then there's hyphomycetes. I don't feel that frightened of hyphomycetes, but I must admit that I don't always get to names. This hyphomycete occurs on Xanthoria periotina again, that yellow lichen and that nitrogen-rich community. It does cause damage to the host, so you can see here the host is bleached and a mixture of colours. It doesn't look very healthy at all. And the apothecia, where the fungus likes to grow, they've gone black and sooty. This is the only Lichenicolous fungus I know of that has an English name. It's called Xanthoria smut, but it's not a smut. It causes this sooty surface, with the soot being composed of small, dark, very ornamented globospores produced from quite chunky hyphae that branch a lot right near the surface of the apothecia, and then they start producing spores just at the surface, so the spores are free to be spread. But I think I've got time to show you my latest really exciting find, which was a hyphomycete. So if you'll indulge me, this lichen in front of you now, it has some dark apothecia, it has some very pale looking apothecia, it has some brown, mottled, dark brown, light brown apothecia. Usually when you find these species, this is a Lecanora species lichen, all the apothecia are roughly the same colour. For each Lecanora, they can vary a bit. So this is a species where you can have quite dark brown apothecia or beige coloured apothecia, but they're normally consistent and they're not mottled in the way that some of these brown apothecia are. Now, I've seen these symptoms so many times and I have never found a cause. So here, the very pale apothecia literally have a missing hymenium. So something's eaten that out. The hymenium's obviously very nutrient rich because it's producing spores. So either some mites or maybe a mollusk has had a good munch and eaten out all that nutritious tissue. So I'm not particularly interested in that. That hasn't been caused by a fungus. But the damaged hymenium, in those damaged apothecia, I thought I'd take a look. Now, this host is one that we might have called Lecanora chlorotera, but recently lichenologists have split that species and give it a whole series of names now. We're beginning to think it's a species complex and we're trying to separate them out. And the way we do that is that we use not ultraviolet, we use polarized light. So the two photographs here have been taken using polarized light. The one in the middle of the slide was a black apothecium which I sectioned. And the one on the right-hand side was a brown, mottled, unevenly coloured apothecium, which definitely looks sick. So you can see that in both of these cross sections, you've got large fluorescing crystals around the outside of the hymenium, below the hymenium and sometimes going up the edge. Those are typical for a lot of species in this group that we've separated Lecanora chlorotera into. But what you've got on the middle photo, the one that's the deeper blue, is a hint of fluorescence across the top surface and descending into the hymenium. And that distribution of fluorescence tells me that that is an apothecia of Lecanora hypocarpa. But if you look at the sick apothecium cross-section, those crystals at the surface are no longer there. Something has destroyed them or prevented them from being formed. Both of these, I should have mentioned, have been stained with ink. So I use a blue-black quink ink, and then I clear with a white vinegar. So that picks out the contents of the hyphae for me. And if we go in a bit closer, you can see those two apothecia again. So in the center, you've got a healthy apothecium. It had the crystals in all the right places, and you can see asci with spores. And because I know the lichen quite well, I knew that those spores were a healthy shape. They were a normal shape. But in the possibly diseased apothecium, I couldn't pick out asci at all. In fact, a lot of the tissue there hasn't even stained. Does that mean it's dead? And what I can see is hyphae growing up through to the surface with spores, many of which have not grown at the tip. Well, they did grow at the tip, but they've now fallen sideways or they've fallen off and they're lying along the surface. I think I've got a photo that zooms in a bit more for you. And here you can see the hyphae coming up, branching slightly, and at the surface they're producing spores. Every one of those spores had 2 cells. They were hyaline before I stained them. And I don't know what this is, but for the first time, the symptoms have led me to a fungus. Now, that doesn't mean that the symptoms are always caused by whatever fungus this is, But I'm sorry, that got me really, really excited. So if you want to look at Lichenicolous fungi and you want to try and get some of them to names, what literature sources have you got to help you? There is a real dearth of books. Until last year, the only book was a Somerset lichens and Lichenicolous fungus checklist. And that's just a list. It doesn't help you identify anything, but it tells you what grows in Somerset. And that's an English southern county, slightly towards the west where it's damper. And if you live in that area, that maybe have helped you because you at least know what's in your area. But last year, the Flora of Lichenicolous fungi, Volume 1, was published by a fantastically hardworking group of mycologists interested in Lichenicolous fungi. And in Volume 1, they covered all the Basidiomycota that are known to grow on lichens across the whole world. It's a marvelous piece of work. It's reasonably technical, but there's fantastic photos in it throughout, a few distribution maps. It tells you where these things have been found across the world. And it's amazing. And the icing on the cake is that you don't even have to wait for your ordered copy to arrive through the post, because there's a PDF version available free online, which you can download immediately and get started on. Absolutely amazing. I can't wait for the other volumes. But it's such a big piece of work that I'm not sure how long waiting time will be. I don't want to get too excited, but I am excited. They're fantastic. It's just a fantastic book. And that's your limit for books, not a lot. After that, another literature source you can go to is scientific papers. The literature on Lichenicolous fungi is spread across a wide variety of journals, many of which are not open access. You're faced with a technical language, which is fine if you're a professional mycologist. It's not so easy if you're a field mycologist. Some of us are just not used to that kind of language. Sometimes these papers are not even in English. I do use Google Translate. It doesn't know the technical words. But an apothecium coming out as a nest, you can kind of imagine that if there's a nest on a fungus, it could be an apothecium. So imagination really helps when you're trying to work out what Google Translate is actually trying to say. Very difficult to find this literature. But Google Scholar is a great help. I don't know how many people have found this website, but this is a search engine freely available through Google. And what it does is it searches the scientific literature. And it searches not just titles and keywords, but it searches within the PDFs that are available to it as well. So it tends to give you a lot of hits. And you can try and look in these papers. But to make life even easier for you, when you search for something in Google Scholar, down the right-hand side, it will pick out for you links to open access papers. So in this case, I've done a search for an Arthonia species, or any Lichenicolous Arthonia, and 3 of those papers have got copies available on ResearchGate. ResearchGate is free to join. You don't have to be a professional mycologist or a professional scientist of any kind. You can sign up for it. Even without signing up, you can go into it, and usually when Google Scholar says there's a paper available, it is available and it's free for you to download. Sometimes it's not the version that got into the journal. Sometimes it's a pre-publication version. So the layout might be slightly different, but you still get all the information you need. Google Scholar is my friend. Other literature sources, you can go online for some of your resources. You can go to the British Lichen Society website. The advantage there is that the material that is available is written in more approachable language. But unfortunately, there are a limited number of species that are being dealt with. There are a couple of host keys, so that if you know the host, you only have to key out the species that can grow on that host, which is really then limiting you to a small group. And often the key will work well and the key is as untechnical as possible. And one of the things that's been done is to combine a host key with descriptions for all the fungi to form a guide. So there's a guide to Lichenicolous fungi occurring on Xanthoria peritinum in the UK. This is now out of date. It's got that problem with describing the host Pycnidia as black, which I need to correct. And it's now lacking 2, maybe 3 species that we've since found. So the guide was written in 2020, and in 3 years we found 3 more, I think it is, which need to be added. But you've got that approachability of language there as the advantage. If you don't mind technical language, the Fungi of Great Britain and Ireland website that's always formed a queue has technical language, but it does cover a lot of Lichenicolous fungi. Doesn't cover them all because this is a voluntarily produced website and no one has time to plug away at it every day of the week. But if the species that you're looking for is covered, you can expect a full technical description, real accuracy, and a good suite of photos, including photos of the disease symptoms on the lichen and also photos of the structures of the Lichenicolous fungus. So a really good place to go. Also online, Dorset Nature is quite good. It has a Lichenicolous fungus section. It has great photos. It's got a cross between very accessible and very scientific language. Again, it doesn't cover a tremendous number of species. It's the species that have been found by one person in Dorset. But it's a useful place to go and covers some of the most commonly found Lichenicolous fungi. Another place to try is the UK Fungi Forum. Anyone can sign up for this. It has a lichens board where people also post about Lichenicolous fungi. It's a chance to ask questions. It is a chance to get help with identifications. People do appreciate it if you do more than put up a photo and say, what's this? They would like you to make the effort to give photos, use a microscope and take more photos if you can, or use a microscope and describe what you see. People would like to know the environment you found the fungus in, which lichen you found it on. All the background information, if you take the time to put that all in, then because you put in effort, people put in a lot of effort. Some of the best experts in the country are on the UK Fungi Forum and are on there frequently. And they will talk to you in accessible language. A really, really useful place to go. And even if you don't want to post anything, just reading the posts, you can learn a tremendous amount. And techniques are discussed as well. So you can pick up tips for how you can work. Another online resource, or 2 more online resources from the British Lichen Society website, are species lists and distribution maps. The Taxon Dictionary on the British Lichen Society website is under the recording tab there. And if you go right down it's usually used for searching, but if you go right down to the bottom, there's a CSV button. It's very small and it's way down the bottom of the page. But if you find it and press it, the website starts collating for you all the data in the Taxon database and putting it into an Excel spreadsheet. And then you're offered the chance to download that. Once you've downloaded it, you've got all the present and past names of lichens and Lichenicolous fungi in the UK. So if you then put a filter on the spreadsheet and you filter for the current name by in the current name column choosing just yes, you can get rid of all those synonyms, and that's helpful. And if you then in the group column choose LF, the Lichenicolous fungus, you will be left with only the Lichenicolous fungi and only the up-to-date names for those. So each fungus will only be represented once. If there's a teleomorph and an anamorph, it will still only be represented once by whichever name is the accepted name. And if it's large enough on your screen, you can see in the little circle that when I did this yesterday, I got 565 species, which seems to be the current number of species for Lichenicolous fungi observed in the UK. The site also has distribution maps which you can access. You go to the recording tab again. If you look down, it says species and maps. And if you click on that, you can search by letter of the alphabet and then zone in on the name that you're interested in. And when you click through to that, sometimes there's some information. Sometimes there is just a map. There's always a map. So you can see here the map for Erythricium aurantiacum, that orange bulbil producing Lichenicolous fungus that goes around destroying Fiscia ascendens and Fiscia tenella in nitrogen-rich lichen communities. And you can see that its distribution is clustered. So it seems to be all over the UK, scattered across Ireland. It doesn't go up right into the north of Scotland and the Hebrides, but it goes almost everywhere else. But the clusters are not hotspots. The clusters are, well, they are, but they're hotspots of lichenologists that look at Lichenicolous fungi. So I could probably name some of those hotspots. And actually, again, I don't know if the cursor is working for you, but if you look at Wales and you go above the peninsula, which ends with Pembrokeshire, and you go up the coast, there's one little red dot on the coast. That's my garden. And the fungus is common on I've got 10 acres. It's common on those 10 acres. I don't know why there aren't more dots in Wales. I guess because I sit at home and read a lot and don't go out walking enough. There are some other lichenologists in the area. They're obviously not sending their records in. It's a fungus that we could find all over the country if we had enough people going out, traipsing the ground and sending in records. And I must point out that these are only records from the British Lichen Society database. At the moment, there is no agreement to share data between the British Mycological Society and the British Lichenological Society. That's not because we disagree about sharing data, that's just because nothing's been set up. If we could put BMS data on there as well, maybe we'd fill in some extra dots. So if I've interested you, or if you're already interested in Lichenicolous fungi, I wish you well finding them. Thank you very much for listening.
And yes, so I just want to say thank you so very much. I'm trying to get my video to come back, and yes, so just thank you so very much. It was absolutely fascinating run through of such an enormous diversity of species. Absolutely wonderful. And no one actually, I'm just looking for some questions. Yes, there is a question here. Thank you. RM, I'm not sure who that is, but RM, thank you so much for the question. So is there a relationship? The question is there a relationship between the type of substrate which the lichen is growing on and the species of Lichenicolous fungus that one finds?
Sometimes, yes. There is often a link between the lichen and what it grows on, and therefore the Lichenicolous fungus, if limited to that lichen, it can't grow on any other substrate. But sometimes there does seem to be a link between the substrate and the Lichenicolous fungus that doesn't involve the lichen being linked to the substrate. Am I making sense there?
Yes.
I can't think of an example, but I am absolutely confident that I've heard of examples.
Thank you. And there's another question from John Watt. Yes, JW, yes, I realized RM was Renos Muzouras, so thank you. I can see your name now. So that the previous question was from Renos, and then we have a question now from John Watts, which is, when it comes to recording, for example, John found Aurinia peltigerina last year. It is on the FRDBI. Okay, so he's put it on there. I think you've just dealt with this question about double recording, but is that something that happens, or does double recording happen? I think that's the question. I hope I've got that right, John.
It definitely happens. The UK isn't very joined up in its recording of biological entities. So people might put something on iRecord or iNaturalist. They might also have access to put it on FDBR, but I can never say that, on the British Mycological Society database. If it was a Lichenicolous fungus, they might send it into the BLS, and there will be duplication. I don't think that's a huge problem. I think it's more important that we get records, but at some point that duplication probably needs to be sorted out. I think providing you put plenty of information in that's connected to your data. So if you're willing to give your name, you're willing to give a good location, you're willing to give a host species, if it's a Lichenicolous fungus, you're willing to put some notes in, so that if the data goes to a different database where they have more fields, they can take information if they've got time out of your notes. So if it's on a lichen, but it's on an oak tree, and you want it to be recorded that it's on an oak tree, the British Lichen Society have a way of doing that. They have a field for that. But if you're going into iNaturalist, iNaturalist has a minimum number of fields, and people tend not to put notes in, which I think is a shame. But it's a quick and easy way that people like to make records. So yes, there's duplication, and hopefully in the future we will be able to amalgamate British wildlife into one big, huge database. The MBN Atlas obviously tries to do that, but I think it has limitations on how well that occurs.
Thank you. That's good. Thank you. Lots of really good feedback. Lots of people have been writing in about how wonderful and interesting the talk has been for them, which is great, and also very accessible, which is lovely. There's a question from Ambikesh. I can't see the full second name for some reason, it's been shortened. So Ambikesh, AD, how can we identify Lichenicolous fungi in the field, or is it just a little bit opportunistic? As in, I suppose if one hasn't quite got the expertise, you know, yeah, I don't know whether that's got your question absolutely right, but I'm just reading from what you've written there, Ambikesh.
It's tricky. I think there are lichenologists who can now recognise Lichenicolous fungi, some of them in the field. There are some that no expert will ever be able to recognise in the field. I think you need, when you start, to take them home and have a really good look at them. If you've got access to a microscope, you can check. So if you find a pink blob on a Fischia species in a nitrogen-rich community, and it's just a blob, and it's pretty bright pink, or even a dark pink because it's dry, if you can take those home, put a blob on a slide, add some water, squash it on a cover slip, have a look under a compound microscope, and you can confirm those spiral spores, then that gives you confidence that the next time you find a pink blob, you might have got the same thing. And by the time you've taken 10 home and you've checked them all and rejected one, but the other 9 were right, then you'll have the confidence to recognise that species in the field. And I think there are many Lichenicolous fungi where you can do that. You can get to know what they normally look like in the field. And then you might pick up something that's a bit different and find out, oh, well, it's the same actually, because it looked different on that day. But that's the only way I think you can learn. It would be good if we had more accessible literature. I actually am the author of what's available on the British Lichen Society website. I did a lot of that post-COVID, but then other things in my life got on top of me, and not on top of me, but took over, and I didn't write anymore. But if we have some more volunteers, it doesn't have to be me who does it. If we have more volunteers, we could expand that. And that was all based on doing identifications with as little technical work as possible. And I think we can do that. If you go to a mycological key in some of the scientific literature, you'll be asked technical questions from the word go. You might come to a couplet that says, are they ASCII? Are they not ASCII? Well, in order to know if they're ASCII, you've got to make it across a squash or a section. But actually, it might be more important, is it pink or black? Which doesn't need a microscope.
Right. Yeah, very interesting. And also what you said there is, you know, the idea that if you go to the effort to use a compound microscope for the first time, and then you get familiar, and then go and look for other ones, but you get a repertoire of ones that you recognize. That's lovely, thank you. Another comment from Gary Easton, just saying that it was a wonderful talk, and he'd like to see more connections between perhaps the British Lichen Society and the British Mycological Society, particularly for perhaps the Fungus Day, but a bit of mutual symbiosis perhaps could be increased a little bit more, and yes, lovely. Then we've got Cam saying he's used the key to Lichenicolous fungi on Vanthoria. Found it really, really useful. So thank you really very much for that. And any idea when it might be updated? So that's from Cameron.
Go and do my work, my day job for a day, Cameron, and that'll give me the day I need to update it. It's not the hugest job in the world. Although if I describe the 3 extra species, it might be. It just depends how easy that literature is to get to. And whenever literature was really, really difficult to get to, I took a private route and managed to get hold of papers. I went to a library, and you can photocopy a small amount of a journal without breaking copyright, so I'd do that. And in those cases, I wrote a scientific description to go on the fungi and lichen of Great Britain and Ireland website, so that the technical description was then available to everyone, as well as the less technical description that I wrote. So it would depend on how much of that work would need to be done as well. I'd love to do it, but I don't seem to find the time.
Well, it's hard to find the time for anything beyond survival sometimes, and it's wonderful that you're doing what it's amazing what you are doing. Thank you so much. But there's a lovely comment from, well, a question really from Leon Fraser saying, are there any examples of extremophile Lichenicolous fungi? That's an interesting question. I don't know what you feel about that, whether you know anything about that.
Extremophile lichens are really, really common.
Yes, of course.
The lichen symbiosis seems to be suited to extreme environments. It's fungi, as well as being, well, I suppose it's because they're good at munching up absolutely everything. Each fungus may be eating a different part of the everything. They're really good at producing chemicals. And it's what we call secondary chemicals in lichens that allow them to cope with these extreme environments, that allow them to dehydrate without harm, that allow them to build their own sunscreen so that they can grow in places where the light is really, really bright and maybe too bright for the photosynthetic tissues and in the algae. And with so many extremophiles amongst lichens, there must be, in the same places, extremophiles amongst Lichenicolous fungi.
Yes. Well, yes, thank you. And another question here is from Ambikesh again. Thank you, Ambikesh. So when I'm seeing the herbarium sample, sometimes a problem is encountered that fungi growing on the lichen may be due to herbarium fungi. How to solve this problem if it is Lichenicolous fungi or herbarium fungi growing due to improper preservation?
You're into a really tricky area there, because you're in that area of molds, of hyphomycetes, where people who study Lichenicolous fungi don't often want to go. Those are the fungi that are least well known. I've found so many acremonium-like fungi growing on really, really disintegrated, dead or near dead lichens. And acremonium is an example of a hyphomycete that has very few characters. You can measure the width of the hyphae that grow across the surface. You can measure the width and length of the conidiophores, but they're very variable. You can measure the length and width of the conidia, but they're very variable in those. You can tell from just one sample that those conidia are variable. And so you end up with very few definite characters. And the acremonium-like fungi are now known to be the hyphomycete, the asexual form of a lot of different teleomorphs. So that makes the taxonomy more difficult because people want to deal with them separately instead of dealing them as one group that you can recognize as soon as you put something under your microscope. So the literature is just not there. I think you'd be back into primary science. You'd be looking at it for yourself, looking at lichens in the field, what grows on those, and then seeing whether the same stuff grows in your herbarium material. Be like a whole primary research area. I don't think there is much to help you unless they are common herbarium specimens that people have written about on plants. And the same species are going onto your lichens.
Thank you. Yes, I mean, presumably that's slightly caused by the fact that when one moves a lichen and changes the environment around it by moving it into any kind of other place where you might be collecting them, or you know, that's going to cause dynamical changes within the relationship and make things more perilous for other.
Yeah, you need to dry them. If you're going to store them, you need to dry them.
Yes.
Not with excessive heat, just like you wouldn't dry a fungus with excessive heat, but you need to dry them reasonably quickly and take away the moisture so that these other fungi can't get in there. And the only thing you're left with were the things that were there when you collected them.
Thank you. Very useful. Thank you. Roy has asked a question about whether any other fungi can grow on the Lichenicolous fungi. So is there anything that might stop another fungus growing on top of a Lichenicolous one? Has that already been looked at, do you think?
I haven't come across any examples of that, but as a plant pathologist, I can think of a lot of fungal pathogens on plants that have their own fungal pathogens. And if it happens there, why on earth wouldn't it happen on lichens?
Exactly.
I've never heard of one.
Thank you. Good question. Thank you, Roy. Nick Shriya has said wonderful talk and was wondering if Polycoccum pulvinatum, another Physica Lichenicolous fungus, is commonly found in the UK? Question mark. I found one easy to identify. I find it easy to identify in the field. So is that one found in the UK? Do you know?
Offhand, no, I don't know how common it is. But one thing you could do is go onto the British Lichen Society website and look at the map. So the map won't tell you how often it's been found, but it will tell you how many hectares it's been found in. And that's a good first call because that data is easily available to you. The other thing you could do is to contact the keeper of the BMS database, a lovely person called Janet, who's done that job for years and years and years. Don't know what we'd do without her. She can be contacted as records at britishlichensociety.org.uk. It's on the website. And you could ask her for the number of records of that species in the UK, or you could ask her for the records and she would give them to you. The British Lichen Society aren't particularly precious with, well, we are precious with our data. We take good care of it, but we're not worried about giving it away. If someone wants to make a living out of it commercially, we'd rather they paid us. But if it's just someone who has an interest in natural history, we are happy to give that data away to encourage more data to come into us, to encourage people's interest. So there are two things you could do to try and answer that question, because I'm afraid I can't answer it for you right now.
Thank you. Thank you for that. P.S. Mota from Colombia in South America is asking a question. Well, just saying that, really a comment more than a question, I think, just saying that he'll not see a lichen community in the same way again after your talk, and your advice and tips were very useful. So thank you. Thank you for that. And then Ambikesh has also said any advice and tips and suggestions how to get started on Lichenicolous fungi. Yeah. Well,
I think despite the fact that there are problems with it, I would download that guide to Lichenicolous fungi on Xanthorrhoea peregrina, because it is an easily recognizable host. It's a widespread host. It's common. It's easy to find the host, and you can look up lots and lots of pictures of it online to see what it normally looks like. And then you can start looking for things that shouldn't be on it. And most of them will be in that guide, and most will key out. So good a place to start as any, and it confines your search. And I think that's always helpful when you're a beginner.
Thank you. D. Napier has looked at the map on the British Lichen Society website for that previous question that came up from Nick Schreier about Polycoccum pulvinatum, and apparently there are quite a few records of this fungus in the UK. So that's useful to know. Thank you, Di, for answering that and for looking so quickly. And that's come to the end of the questions in the chat. So I've got a couple of short questions myself. One is about the way in which the Lichenicolous fungi may, well, it's just a question whether they alter the nutrition that animals might find in lichens, because we know that, for example, reindeer, I think, caribou often graze on lichens and even scratch the snow, don't they, I think, to get hold of lichens. So I wonder whether Lichenicolous fungi would damage or alter drastically, or even make fungi that lichens that are normally quite good to eat quite poor.
I think you've got me back into the logical guesses there, Christian. I've got no idea if anyone's worked on that. It's another really interesting area of biology, isn't it? Which I wouldn't want to know about. I mean, I'd quite like to hear the answer, I suppose, but I wouldn't like to put in the work. I would guess yes. The contents of that lichen are going to change. Some pathogens would eat the readily available nutrients. Maybe that's why some people go straight. Some fungi go straight for the algal partner. Mm-hmm. I would guess yes.
Yeah. Okay, great. Thank you. And another question I have is about the little black lines that you sometimes find between adjacent lichen colonies, which I believe is a kind of pseudoscrotal plate, which you often find between, for example, basidiomycete and ascomycete fungi inside spalted wood. And my question was, well, just around whether the Lichenicolous fungus entrance into the dynamics might alter the thickness or the outcome of that line, that territorial marker.
Well, I've got a guess for that one as well, and that would be that Lichenicolous fungi would stay away from that area. Mm-hmm. That area is laid down to stop the fungi overgrowing each other. Neither fungus wants to give another fungus their own territory. They don't give their territory up. And so I imagine that the chemicals that are pumped into that black line are not nutritious, are not easily digested, and would not interest a Lichenicolous fungus.
No.
But that is pure guesswork, and I'm willing to admit it.
Great, thank you. That's lovely. Final question from me then is just, well, I suppose it has to be asked in light of all the news about the overheating planet, whether there are any changes that you could guess might happen that to the Lichenicolous fungal communities due to the climate changes that are happening.
There are definitely things happening. There's lots of things happening with lichen distributions in the UK as well. A lot of species that were confined to the south that are spreading north. There are changes in the Lichenicolous fungi flora, even in the few years that I've studied them. So the Athelia arachnoidea, it really does seem to be rarer. I don't find it very much anymore. And when I have found it, I found it in Wales. Previously, I was finding it in Bedfordshire. Now I can't find it in Bedfordshire. That has this exposed cobweb of hyphae, so maybe it just needs a more regularly moist air to keep it going. But it's definitely not as prevalent in the southeast as it was. And the other change I've noticed is that on the Fischia species in that nitrogen-rich community, I used to see a lot of Elosporiopsis christianseni. And there's a lot of debate as to whether that grows on bark or it grows on lichens, whether it harms the lichens or whether it just eats them when they're harmed already. And that debate's not been solved. But it used to be really common and really easy to find. And it is now less common. And the Erythricium aurantiacum, the orange bulbil forming fungus, that is more common. So it's as if that same niche is being taken over by a different Lichenicolous fungus. So both existed, both were in the UK, but has the balance between them changed? But is that anything to do with climate change and climate warming? Is it to do with the rain patterns that have also changed? Is it to do with the nitrogen concentrations in the atmosphere? I can tell you these changes are happening, but I can't tell you the reason.
Thank you. Well, it's very interesting. Yes, I suspect that it's very difficult to disentangle all those different types of variables. But very interesting that you're seeing those changes. That's fascinating. And we are seeing also, well, in Devon, for example, we're seeing completely new species of fungus arrive. Some basidiomycete fungi, which is orange. I think they call it the orange ping pong bat fungus, which is a
so you're right in the south coast there, aren't you?
Yeah.
So you get things first.
Yes, it's arrived, and it's quite common actually in some little woodlands near the Dart Valley. So things are definitely changing. Well, thank you. It was a fascinating talk, and lots of people have been writing and just saying thank you so very much. And I'd like to thank you so very much on behalf of the British Mycological Society for giving such a wonderful presentation full of the biodiversity of such an interesting group of fungi that obviously, you know, we don't really hear enough about. So it's been very, very interesting indeed. It's really just for me to say thank you on, also to the audience for coming along tonight. So thank you everyone who's been attending and for your excellent questions. I think really it's probably time to say goodbye and have a really wonderful rest of your evening for everyone. Yes, thank you so much again, Fay, for such a wonderful talk.
You're very welcome. Good night, everyone.